If you want to live a happy life, tie it to a goal, not to people or things, and struggle all this to get your goal rather than to fight with the people. This is the saying of the Albert Einstein. So the continuing the exotoxin and endotoxin, first the exotoxin, diphtheria toxin or the toxin of the Corynebacterium diphtheriae. These bacteria carry a temperate bacteriophage with structural gene for the toxin. These are now toxigenic and produce diphtheria toxin under the influence of the temperate bacteriophage. Otherwise, these bacteria are not toxinogenic and now they become toxinogenic and produce diphtheria toxin. This toxin is enzymatically degraded into two fragments, A and B, linked together by a disulfide bond. Both fragments are necessary for the toxin activity, as I have already shown you in the previous slides. A and B linked together. The tetanospasmin or the toxin of the Clostridium tetani. Clostridium tetani is an anaerobic gram-positive rod that produces spores. It contaminates wounds and the spores germinate in the anaerobic environment of the devitalized or the crushed tissue of the human being, and the vegetative form produce toxins, tetanospasmin. It reaches to the central nervous system by the retrograde transport along the axons and also through the systemic circulation. Very small amount of the toxin blocks the release of an inhibitory mediator in motor neuron synapses. So, if the release of inhibitory mediator is there, so the result is localized, then generalized muscle spasm as the inhibitory mediator is blocked. Extremely small amount of the toxin can be lethal for the human beings. Here one can see the diphtheria toxin, and this diphtheria toxin under the influence of the bacteriophage, that bacteriophage transmits the genetic material inside the bacteria, and this A and B toxin activates, and that is just going to cause the cell death because of this bacteriophage induced toxigenicity. By these just prevent the protein synthesis by the ribosome, and the ultimate result is the cell death. Now, about the tetanus toxin, the mechanism of tetanus toxin is this Clostridium tetani, as you can see that it is going to produce tetanospasmin. So, at neuromuscular synapse, as one can see that excitatory transmitters are being released, but because of this tetanospasmin, these tetanospasmin are just going to attach with the inhibitory transmitters, and these inhibitory transmitters are blocked, and that is going to cause the inhibitory transmitter release. Blocking is just going to cause the continuous stimulation by the excitatory transmitter which is being released, and the inhibitory transmitter which is here, that is not able to reach to counter this excitatory transmitter, as it have been blocked by the tetanospasmin here at the synapse. So the result is continuous stimulation by excitatory transmitter. Now, botulotoxin or botulinum toxin, this bacteria is found in soil or the water, grows in foods in anaerobic environment. A most potent toxin is produced, which is heat labile but need high temperature. There are 8 serological types of it. Type A, B, and E cause the human disease. Mostly, toxin is absorbed from the gut and carried to the motor nerves. Here in the motor nerve, it blocks the release of the acetylcholine at the synapse and the neuromuscular junction, so the muscle contraction does not occur, resulting in paralysis. The other toxin is the toxin of the Clostridium perfringens. The spores of the Clostridium perfringens are introduced into the wound by the contamination with soil or the feces. In presence of the necrotic tissue and anaerobic environment, spores germinate and the vegetative cells produce many toxins. Some are necrotizing and hemolytic and favor spread of the gangrene. Alpha toxin is a lecithinase that damages the cell membrane. Theta toxin and other toxins has a necrotizing effect. Here one can see first the botulinum toxin. Mechanism of action is Clostridium botulinum. It is going to release the toxin. As you can see, these toxins are going to attach and hinder or block the release of the acetylcholine from here, and the result is the acetylcholine is being blocked by these toxins and motor end plate is not able to work. So the toxin blocks the acetyl release, and ultimately acetylcholine is responsible for the stimulation of the muscle fiber, and the stimulation is blocked. So ultimately the result will be because of blocking of acetylcholine vesicles by these toxin of botulinum. Now, the toxin of Clostridium perfringens, as one can see, these toxins are going to bind with the surface, then they are going to insert inside, and then these are going to disrupt the whole cell, and ultimately the result is the destruction or disruption of the system. Now, streptococcal erythrogenic toxin, some strains of the hemolytic streptococci produce this toxin that produce a punctate maculopapular erythematous rash, as in case of the scarlet fever. Production of this erythrogenic toxin is under genetic control of the temperate bacteriophage, as it was in case of the Corynebacterium diphtheriae. If phage is lost, streptococcus cannot produce this toxin. So this is all toxin production under the control of temperate bacteriophage. The next 6 is toxic shock syndrome toxin 1, TSST1. Some Staphylococcus aureus strains growing on the mucous membrane of vagina during menstruation or in wounds elaborate TSST1. This causes toxic shock syndrome by unknown mechanism. It is severe to fatal problem even with the antibiotic treatment, and the patient cannot recover because of the fatal effect of this toxin. There is shock, high fever, diarrhea, vomiting, and diffuse red rash involving many organs of the body. Here one can see the Streptococcus erythrogenic toxin. It is have got the 2 chain, alpha chain and B chain, and that is just going to enter into the membrane, and through transmembrane region it can enter into the cytoplasm of the cell and causing all these destruction and problem. The toxic shock syndrome toxin, as one can see, these are the exotoxin released by the staphylococcus, and these are going to cause the different effect on the host cell. So these are just the toxin TSST and other toxin like this causing the damage, and the specialty is toxic shock syndrome. Exotoxin associated with the diarrheal disease, these are enterotoxin, toxin produced by the enteric pathogens like E. coli, Vibrio cholera, Staphylococcus aureus. Enterotoxin is also the exotoxin. Other enterotoxin produced by the strains of Yersinia enterocolitis, Vibrio parahemolyticus, and Aeromonas species are all the exotoxin. Now, the other variety is endotoxin. We have talked about the exotoxin released by the cell cytoplasm into the medium and affecting. Now, the endotoxin. The term endotoxin was cloned in 1893 by Pfeiffer. It is a class of toxic substances released after lysis of the bacteria. So it is the component of the gram-negative bacteria. Endo means inside, not secreted by integral part of the cell wall of the gram-negative organism only. The classical and most potent endotoxin is lipopolysaccharide, which is the LPS. Certain peptidoglycan of certain bacteria also displays many endotoxin-like properties. So here one can see the cells, the cell wall is just going to be destroyed, and these are behaving like the endotoxin. So these are the just the part of the cell envelope or the cell wall of the gram-negative bacteria. These are liberated in blood when bacteria do die and the lies. The substances can be extracted in the laboratory with the phenol water, and these endotoxins are heat stable at 100 degrees centigrade for 1 hour because heat stable, no toxide formed like exotoxin, so no vaccine can be made because of this heat stability. No effective therapy to worsen the toxic activity of lipid A or the peptidoglycan, which behave like endotoxin. These are toxic only in the high doses. Endotoxin are poor antigenic and weakly immunogenic, toxicity not completely neutralized by the antibodies as it is in case of exotoxin. These endotoxins do not exhibit specific pharmacological activities as in the exotoxin. The pyrogenic effect of the fluid used for the intravenous administration is due to the presence of the endotoxin from the contaminated bacteria. So these are going to cause the fever because of this contamination. These endotoxins exert effect by stimulating macrophages to release cytokines at very high level. The picture says a lot about the father's effort, but the father's effort is hidden, and the kid only sees the mother love, and that is the mother is just eyes are in front of the child, and the child is not able to see the hands of her father, which has given the support to the child, and the legs of the father who has given the support to the mother. So the mother is able to do love to the kid, but the appreciation is always of the mother, and the fathers are always the forgotten heroes. Now, basic structure of endotoxin. Alternate name is lipopolysaccharide, a molecular complex of the lipid and the polysaccharide. Mostly endotoxin molecules are similar in chemical structure and have nearly the same biological activity. The molecular complex is divided into three regions: O-specific chain with many repeating oligosaccharide residues, core polysaccharide, that is the backbone of the macromolecule of endotoxin, and lipid A is composed of the glucosamine disaccharide with attached long chain fatty acid and phosphate, which is the main causing the problem and the lethal weapon of endotoxin. Here one can see the gram-negative bacteria. It have got cell membrane, periplasmic space, peptidoglycan, and this outer membrane, lipopolysaccharide, green color, behave like the endotoxin. Similarly, one can see the gram-positive bacteria and the outer membrane of the gram-negative bacteria, that is the what behave like the endotoxin that is not present in gram-positive bacteria. So that is the basic structure of endotoxin. It have got three parts: the O antigen, that is the outer part, and outer core, and the central part is the core polysaccharide, and then there is the lipid A part, which is mainly causing the damage, and that is the toxic part of the endotoxin. So polysaccharide part causes antigenic diversity. Lipid A moiety causes the toxicity. Members of the family Enterobacteriaceae exhibit O-specific chain of the various lengths, whereas Neisseria gonorrhoeae, Neisseria meningitidis, and B. pertussis contains only core polysaccharide and lipid A, and they do not contain O-specific chain. So these are called lipo-oligosaccharide instead of lipopolysaccharide. Biological activities of all endotoxins are same, but some are more potent than the others. Now, the biological activity of the endotoxin, these exert effect by stimulating macrophages and getting the release of cytokines like IL-1, and TNF, which are responsible for causing damage to the host cell. Lipid A cause inflammation and tissue damage, activation of B cell clones. Most of the effect of the endotoxin are mediated by the tumor necrosis factor, that is TNF. Attempts are now therapy using anti-endotoxin or anti-TNF antibodies to neutralize the effect of tumor necrosis factor. Endotoxins are non-specific inciters of the inflammation, and the biological activity of the endotoxin is they are capable of producing fever, pyrogenicity, leukopenia, decrease in TLC followed by increase in TLC, that is leukocytosis, hypotension, decrease in blood pressure, hypothermia, low temperature, gram-negative shock, endotoxic shock with dirty intravenous equipment like trips and other things which are used for IV line, life-threatening complication of the septicemia, that is in the meningococcal infection, decreased peripheral circulation and perfusion to the major organs, that is causing multiple organ system failure, the effect of endotoxin. Now, the next effect is activation of the C3 and the complement cascade, stimulation prostaglandin synthesis, activate kallikrein system, kinin system, clotting system and fibrinolytic pathways are also activated, mitogenicity, potent B cell mitogens, disseminated intravascular coagulation, sepsis and lethal shock, death, peptidoglycan, teichoic acid, and the lipoteichoic acid of the gram-positive bacteria can also produce endotoxin-like toxicity, as I have told you. So these can behave like endotoxin, but they are basically the exotoxin, but behaving like endotoxin. So here one can see the effect of the endotoxin on the histamine, tumor necrosis factor, activation of the coagulation system, prostaglandin, thromboxane, leukotriene, and prostacyclin release, endorphin release, bradykinin, oxygen-derived free radicals, platelet activating factors, anaphylatoxins like C5A and C3A, myocardial depressant factors, interleukin A and interleukin T, causing the fever and the other systemic feature of endotoxin. Now, why the endotoxin are beneficial to the host? It is quite strange. They are causing damage, but still they are of some benefit to the host. These are mitogenic effect on the beta lymphocytes that increase resistance to the viral and the bacterial infection. Induction of the gamma interferon production by T lymphocyte act as the antiviral state in case of hepatitis. Promote rejection of the tumor cells and activate macrophages and natural killer cells, activation of the complement cascade with formation of C3A and C5A. So the killing effect of the other bacteria also induce formation of interleukin 1 by macrophages and interleukin 2 and other mediated by T lymphocyte. So these are all the benefit of endotoxin. It's just unbelievable. One can imagine that 2% in one photograph, and it is really the masterpiece of photography. and the art which the artist have made. Unbelievable. Now, detection of the endotoxin in the medical solution, how we can detect it. Endotoxin is omnipresent in environment, found in the deionized water lines in the hospital and the laboratories. Try to avoid exposing patient to the medical solution which are containing and contaminated with endotoxin. Though all supplies are sterile, contamination occurs due to the improper handling of these things and having the endotoxin. Water used in preparing such solution must be filtered through ion exchange resin to remove the endotoxin completely, and because autoclave sterilization or the filtration through bacterial membrane filter is not effective. Suspected endotoxin containing solutions are checked by the two procedures, one by the rabbit pyrogenicity test and other by the litmus lysate test. The rabbit pyrogenicity test is based on good sensitivity of the rabbits to the pyrogenic effect of endotoxin. Test sample is injected intravenously into the ear veins of the adult rabbits. Now, rectal temperature of the animal is monitored and a sensitive and reliable indicator of presence of the endotoxin because they are going to increase the body temperature of the rabbit, and that is the pyrogenic effect of the endotoxin. Here one can see the rabbit pyrogenicity test. The endotoxin is being injected into the ear vein of the rabbit, and then rectal temperature is checked to see the endotoxin activity. The other test is limulus lysate test. More common, it's less expensive and simple and fast also. Endotoxin do induce gelation, freezing of the lysates of the amoebocyte cells from the horse, shoe, crab, litmus, polyphemus. Very sensitive test, 1 nanogram per ml, that trace quantities of the endotoxin can be detected by this method, give rapid result in CSF, but not in the blood, as the blood contains inhibitors that prevent gelation, so not effective for the blood samples. Test kits are commercially available, and the other samples can be tested by limulus lysate test. Here, that is the limulus test, that is the kit of that, and one can do it. Now, the summary of all this exotoxin and endotoxin is: exotoxins are the proteins, while the endotoxins are the lipopolysaccharides. Exotoxins are the actively secreted by the live cells. Endotoxins form integral part of the cell wall, released by the cell lysis, only released when the bacteria die and the cell wall disintegrates, so the endotoxin is produced. These exotoxins diffuse into the surrounding medium, but the endotoxin do not diffuse into the surrounding medium. They go themselves to act on the targets. These exotoxins are produced by both gram-positive and gram-negative bacteria, but endotoxins are produced only by the gram-negative bacteria. Exotoxins are the highly toxic and fatal in minute quantities. These are of less toxicity or the moderate toxicity, the endotoxin, and the active only in very high and large doses. And exotoxin have specific tissue affinities like blood, liver, and nerve. They act on these parts of the body, but endotoxin are non-specific action of all endotoxin, and they cause systemic effect and fever. Action of the exotoxin often enzymic, but the action of the endotoxin not enzymic action. Exotoxins are usually do not produce fever. The endotoxin produce fever by the release of interleukin-1 and TNF. Exotoxin have specific action of each exotoxin, and that is acting on the different body tissues. But endotoxin have no specific action and tissue affinity that can affect on any tissue. Exotoxin are heat labile, inactivated at 60 to 80 degree temperature, but endotoxins are heat stable. So, due to this heat labile, they are able to produce toxide and not able to produce toxide because of the heat stable. Exotoxins are highly antigenic and endotoxins are weakly antigenic. And exotoxin stimulates antitoxin antibody production. The endotoxin not stimulate antitoxin or cause antibody production. Action of the exotoxin can be neutralized by the antibody. Action of the endotoxin cannot be neutralized by the antibody. Exotoxin readily separable from the culture by physical means like filtration, and endotoxins are only obtained by the cell lysis, and exotoxin frequently controlled by the plasmid, that is extrachromosomal genes, but endotoxins are synthesized directly by the chromosomal genes. So that is the end of the lecture, and I hope you have liked it, and I request to just subscribe it and do share it with others. And if you have any question or any query, please just ask me, and I hope you have enjoyed this lecture and you will tell your colleagues and others about this. Thank you very much for with me